So, welcome to a special seminar that is both the regular Ecology and Evolution seminar and also a special lecture in the Major Issues in Modern Biology series sponsored by the Storer Foundation. We're delighted to have as our guest today Professor Naomi Pierce from Harvard University, a very apt choice for this special series. I'll try to make the introduction brief. Naomi grew up in, I never got to ask her, was it Colorado?
Yeah.
Colorado, got her PhD at Harvard. Correct me if I'm wrong, had early appointments at Oxford and Princeton, and then joined the Harvard faculty, I think, in 1990, where she is the Hessel Professor of Biology in the Department of Organismic and Evolution Biology, and also Curator of Lepidoptera in the Museum of Comparative Zoology. Naomi strikes me as one of these people who is an extraordinarily determined and intrepid explorer of biological diversity. I first met her when she was in the field in Australia, more decades ago than I'd like to think. And since that time, I've just been impressed with the extent to which she has broadly sampled taxa and systems of interest to her. In a broad sense, her research is concerned with the ecology and evolutionary history of species and species interactions, but with a special emphasis on butterfly ant-plant associations, which range from mutualistic to parasitic, and also more recently with insect-microbe symbiont associations. And what characterizes much of this research is a willingness to delve into diverse, multi-species systems where the relevant players are often scattered all over the globe. And that has not stopped her one bit from getting the relevant material, whether it's from Australia or South Africa or Iran, as she was telling me earlier today. She's been tenacious in getting appropriate taxon sampling, so that the result of her is that her research is imbued with a rich sort of evolutionary context for understanding the species interactions that play out on an ecological timescale. The system she'll talk about today, if I'm not mistaken, includes a group of butterflies that were of particular interest to Vladimir Nabokov, who, as it turns out, was not just a famous novelist, but also someone who dabbled in butterfly taxonomy. And maybe even dabbled is an unkind word, because it turns out that some of his findings and speculations were quite prescient. And that's really Naomi's story, not mine. So without any further ado, please join me in welcoming Naomi Pierce.
Thank you very much. It's a real pleasure to be here. Okay, so my title today is Nabokov's Blues. I chose that title when I was talking to Phil, and I heard it was a public lecture. Later, I heard it was Major Issues in Biology. And of course, it may not, on first glance, think that Nabokov's Blues are going to lead to major issues in biology, but I'll do my best, because as I've found over the years, these butterflies are nice exemplars for many different aspects of biology, and I've tried to choose a few that would interest a rather broader audience. So, for many years, my students and I have studied the lysinoidea. These are the blues, coppers, and hair streaks, and also the metal marks. So the rhyodinids and the lysinids together are the lysinoidea. And we've studied them largely because the caterpillars of the majority of species associate with ants. These associations range from mutualism to parasitism. They're facultative. In North America, most of the associations we see are facultative ones. So we're not so used to the very elaborate obligate ones you see in the southern hemisphere. It's been a very rich a rich ground for exploring species interactions of all kinds. But that is actually not what I'm going to talk about today. I'll have three sections to this talk. The first is a very whiz-bang tour through the lysinoidea, just so that you can see them and I can show off a phylogeny that we've recently, or not so recently, finished for them. And then I'll talk specifically about the Poliomatis blues, which are the butterflies that Nabokov was particularly interested in. And I'll give you the two sides of the coin, which sort of historically have been why my lab, or why we've worked on them. Okay, so first, just for the lysinoidea, which include the lysinidae and rhyodinidae, I will go back and forth between them because I tend to think of them all as lysinidae. This is the state of the art when I started working in the field, in this field. Colonel John Elliott had assembled a phylogeny for the the Lycaenidae based on morphological characters. And it was a nice, he called it a tentative arrangement. I really had no idea how good this phylogeny would be when I started out working in this area. So I rapidly started gathering taxa and working on a molecular phylogeny to complement his morphological one here. And something like 800,000 base pairs and 10 years of collecting and much sweat and tears later, this is what it sort of looked like. And this is our mantra for the lab. Here's the phylogeny of the Lycaenoidea. I won't go into the gruesome details except to say that it's very well supported with something like 8 or 9 nuclear genes and 136 morphological characters, largely of the adults, used to infer the relationships between the groups. And there was quite good agreement between the molecular phylogeny here and the morphological phylogeny here. The one thing that sort of jumped around was the position of Lepharis and Curatus, these funny enigmatic groups that consist of just a few species that are kind of at the base. I would say Curatus is sort of that missing link between the Lycaenids and the Rhyodinids. So just to give you a feel for the rich biological diversity of this group, the Rhyodinids are largely distributed in the neotropics. They have the greatest variation in wing shape and size of all the butterflies. And here's a closer look. Here we're just zooming in on a part of the phylogeny. You can see almost all of them are neotropical here, with a small group Old World clade. And Styx infernalis was always thought to be the missing link. Turns out not to be. In fact, it's an Old World one, which is rather surprising. It's distributed only in the New World, or its closest relatives are Old World. Here, Curatus, our funny 18 species in one genus. It's our funny missing link between the Rhyodinids and the Lycaenids. And at the base of the Lycaenids are this Aphneine. This group is largely South African in distribution, and a great many species are carnivorous, as we're discovering the more work we've done on them. They're very closely allied to the Militini. All the species in the Militini are aphytophagous, or I'll say carnivorous, but that means eating anything other than plants. So this is a big radiation of that trait of carnivory in the butterflies. It's the caterpillars, of course, that eat either ants or insects associated with the ants. Closely allied to the Parisiini, whose life histories are hardly known, although there is one group that has a sort of tent caterpillar-like behavior, doesn't seem to be ant-associated. Here they are. This was just zooming in again on this basal earliest clade within the Lycaenids. And it was a big surprise. The Aphneine had previously been put in quite a derived position, but Eliot had even gone back and changed his tentative arrangement, put a little insert into the monograph he wrote saying, I can't decide if they're really basal or they're really derived. And so he, as it turned out, really knew his stuff. The trees that we got were a very good match to his intuition about almost all of these groups. We move on then to the Lycaeninae, from which the Lycaenids get their name. These are the largely coppery species, although there's a nice Old World Heliophorus group. And the Theclines, a huge group of Lycaenids found worldwide. These are have the hair streaks, these characteristic fake head spots and almost antennae-like projections. So here you can see the Theclines. Here with lots of interesting surprises, various things that moved around, but I'm in the taxonomy, but I won't dwell on it because I don't have time today. And then the Polyomytini, the last vast group within the Lycaenids, which Eliot threw up his hands at actually even giving a structure to the tribes and so on of Polyomytini. Well, there were a couple of tribes, but he threw most of them into sections, and he just said, can't tell between them. There are all these different sections. These are the groups. These contain the blues, which I'm going to talk about, the blues that were so much the focus of Nabokov's interests. So the Glaucopsyche lignimus, many of you may know, is a local species found around here. So there are the Polyomatini in our last group, and some resolution for that group. Now, of course, I did this phylogeny originally, my students and I, because we wanted to do comparative life history analyses. There were two things that came out of this. One is that it's hard to do the kind of comparative analysis that you want to do of life histories when you have what is really a placeholder phylogeny, so that any one of the taxa here represents maybe, anywhere from 100 to several hundred species, all of which may have varying life history characters. So on a broad brush scale, you can do something. You can look at certain patterns. I mean, the biogeographics, just for fun, I've pulled this out. You can see here that there's a big neotropical clade. The rhyodinids are neotropical, and that most of the diversity of the group is African. The basal groups are African, and most of the diversity is in the Oriental region. You can do some really large-scale things. We sort of had fun thinking about aphytophagy. This is something that fascinates us, I suppose, because 99.99% of all Lepidoptera feed on plants. There are very rare instances of aphytophagy in the Lepidoptera as a whole, but many in the Lycaenidae. And actually with a very interesting distribution where what we frequently see are many single evolutions of the trait of carnivory, largely eating ants or insects associated with ants, and only a very few radiations in the group. To study this, we really went down at a different level. We looked at things like, here's the evolution of cuckoo feeding in Maculinea. This is a group, a genus that's largely distributed in Palearctic. And there are a few taxa that actually they tap on the ants' mandibles and are fed mouth to mouth by the ants. And we could ask a simple question: is this a derived feeding preference within that genus? Is the simpler thing to just eat the ants directly? And for this, we went and collected all these taxa. This phylogeny was published in Nature. I sometimes wonder why, to be honest, except that Maculinea it is of enormous conservation interest in wherever it occurs, because these butterflies have such complex life histories where they start off feeding on flowers, and you can see they're tracking. The phylogeny tracks very well, very conservatively, the flowers that they feed on initially, and then drop to the ground and go into the ant nest, where they either eat the ants directly or are fed mouth to mouth by the ants. And one simple, in a simple comparative way, we can say, well, no, it's certainly not a derived trait, at least in terms of the polarity on this tree, this being cuckoo feeding. It's a bit of a hand wave, because it turned out that there was another genus, Fangaris, that Maculinea was nested in, and the full life histories of that group aren't known. But I hope it gives you a good flavor for the kind of work that we tend to do in the lab. Since I don't have much time to go into ecology, I thought I'd throw that in there. Okay, so what the focus of my main interest today are on the butterflies that Vladimir Nabokov particularly loved. And he loved the blues, and he loved those in the section Polyomatis. Now, quite by chance, I had encountered Polyomatis long before I paid much attention at all to what Vladimir Nabokov had to say about Lycaenid butterflies. And just to show you where we are, there's the Lycaenoidea again, and here we're going down to the Polyomatini, and Polyomatis is just one section that occurs right in here. Okay, this right in here. It's just a small piece of the diversity that we've looked at already. Now, this story started when David Haig, one of my closest colleagues at Harvard, came into my office and said, well, all this ant association in the Lycaenids is okay, Naomi, but why don't you work on the interesting species? Why don't you work on the ones that have strange chromosome numbers? So David is a theoretician and a very good one, very insightful one, and I usually listen to what he has to say. He's Australian, and he had been much influenced, as many Australians are, by M.J.D. White, who had written widely about speciation, but was particularly interested in the role of chromosomes in speciation. Here's a picture taken from his book, Modes of Speciation, showing that for Lepidoptera, the modal chromosome, the median chromosome number is about 31, and for Lycaenidae, it's about 24. But see this tremendous tail, this great range over here. These are all Lycaenid butterflies. And he comments in there, you know, that this is a very unusual thing, and in fact, it's very poorly understood. So I started tracking this at that time. I went back and I looked up some of these records, and there were many different names, but eventually they all, you know, they all fell into just three genera, all in the Polyomatus section. This Agrodioides, Lysandra, and Plebicula. These three genera have the widest range in chromosome number in the animal kingdom, ranging from n equals 10 up to n equals 225. So spectacular range in chromosome number. Now, chromosome number and how you package your genome is very interesting. It's easy to understand why M.J.D. White was so fascinated by the role that it might play in speciation. I mean, karyotype is a key character involved in species isolation, because if you have different chromosome numbers, and generally this is a trait that's highly conserved, but if you have different chromosome numbers, you can have reduced fertility in the heterozygotes, or just a straight out selection against meiosis. You can't undergo meiosis if you've hybridized with different chromosome numbers. You're in big trouble, and reduced recombination. There are lots of problems, and as Jerry Coyne and Alan Orr remarked, they said it's surprising, remarkable that no one has systematically studied the number of chromosomal rearrangement differences between taxa as a function of divergence time, in here measured molecularly. I think they put that in a slightly different way. No one except them, actually. The best example for approaching this and looking at this kind of question was a paper that they published looking at the relationship of Drosophila to each other with respect to chromosome number. Well, David suggested that we work on this. I sent a student to Spain. I saw that there were at least a dozen species in Spain of this genus Agrodioides that had very widely divergent chromosome numbers, and I thought that would be a good place to start. We could map them on a phylogeny, and it would be interesting. And at that point, an email came for me from this fellow, Nikolai Kandel. Nikolai Kandel had gone to a meeting where he'd met one of my female graduate students, and I don't know what he exactly wanted, but he was writing to me asking for her telephone number. And I looked at the name and I thought, Kandel, I've seen that name before, and I remembered that he had published a paper on chromosome numbers in Agrodioides, and he was coming from St. Petersburg University. I thought, well, great. I wrote back and I said, oh, did you work on Agrodioides? And he wrote back and he said, yeah. He said, I'm just finishing my master's degree on these, and I'm looking for a place to do a PhD. So this was actually very thrilling for lots of reasons. So he came to Harvard, and sort of with him came a great collaboration with his master's advisor, Vladimir Lukhtanov, from St. Petersburg State University, whose great love in life are these Agrodioides butterflies. And Lukhtanov is a master at karyotype. Getting insect karyotypes is not an easy business. It's a real art form, and he's great at it. Okay, well, of course, there are lots of interesting questions to be answered. You know, what is the role of chromosomes in speciation? Are the patterns of speciation common for different organisms? There is reinforcement, and I'll come back to why reinforcement is so interesting, a common mechanism of speciation. Why do you sometimes get cryptic ones, and why sometimes morphologically distinct ones? And how and why do morphological differerences between species arise? These are just some of the questions that immediately come to mind when you think about a group of organisms that have such divergent karyotype or chromosome number. And the genus Agrodiotes was an absolutely marvelous model system for looking at this. It has this wide Palearctic distribution, many, 120 taxa in this Western Palearctic, with most of the diversity, and here is the slight drawback, most of the diversity is in Iran and Eastern Turkey. It was difficult. And you get many species with allopatric distributions, these dot-like distributions, but in some areas you get, you know, as many as 20 different taxa all co-occurring at the same time. Now, the males of the majority of these butterflies are blue and very consistent with each other, as you can see here. But in some locations, they vary rather considerably in the hue of their wings. So you get this fascinating range in the hue of the male wing color. And that turns out to be important. I'll come back again, because we all know wing color is, butterflies use wing color in mate recognition and other forms of what pre-zygotic, before the zygote forms, reproductive isolation. And as I said before, they have this spectacular karyotypic diversity. And this is a plate from one of Vladimir's papers giving karyotype information about Agrodiotes. What I've added here to remind myself to say this is not a case of polyploidy. As you can kind of see if you squint at these are 14 different karyotypes, and this sort of gives an idea of how the plate, as you look down, what it looks like when you're studying these. But the overall genome size is roughly similar between these different species, but the variability that we're looking at is generated by either fusion or fission of these chromosomes. So fusing when you have a few number, and fission when you have the very high numbers that are often very hard to count. Well, what followed from there was an awful lot of field work, largely undertaken by Vladimir and Nikolai, and Roger Villa, who I'll talk about later, Rod Eastwood in my lab. The field work was essential because we had to collect the butterflies. You collect them in the field, you whip off the testes, you fix the testes so that you can then look at the karyotype, and then you pickle the rest of the insects so that you can be sure that the DNA that you're sequencing matches the karyotype that you've gotten from that individual. So there was a lot of collecting involved. 4 expeditions to Iran alone, which were entirely led by Vladimir. Okay, and here it is, the phylogeny of Agrodiotes. Nikolai sequenced like a it was like the military in my lab. He was there day after day after day sequencing and produced this fantastic phylogeny very quickly. You can see that the traditional morphological groups, which are in different color patterns, none of them were completely upheld, although there's still there's some rough correspondence. But one of the things that was very exciting, we could hardly wait till we got this phylogeny, because we wanted to map the chromosome number onto the phylogeny and look at how chromosome number varied over time. And we did see some cases of saltatory change. So one sister taxa pair where this species is n equals 10 and this species is n equals 80. But in general, if we took each one of these clades and we looked at and we looked across the clade at here I'm calling it nucleotide divergence, but you could also call that time, and we looked at the mean karyotype difference, what we found was that chromosome number is gradually changing over time and changing away, forward in higher numbers and lower numbers over time. And one way that I like to think about this is if you take two populations of these butterflies and you separate them allopatrically from each other, so they're in different places, now their chromosome numbers are gradually going to start to change from each other, away from each other. Okay. So we did this phylogeny, and at the same time we noticed a very interesting pattern, and that is that when you collect the butterflies in the field, if they're located in the same place, so sympatrically, the males tend to have different hues to their wings. Whereas if they're located allopatrically, these sister taxa tend to have the same color. Okay, so this is a snapshot of the general pattern that seemed to be occurring across this whole phylogeny. Sister taxa that live in different places, the males are the same color. Sister taxa that live in the same place, the males are different in their color. And that played out across the whole phylogeny. Here you can see different hues to the color, which we had people come in and score these subjectively because that seemed to be the fastest and easiest way to measure it. And I remember, and I think this will just show you the sister taxa. You see how many pairs we were able to compare up and down this tree. And we sent it off. We were thrilled with this result. We immediately sent it off. The paper wrote it very quickly, and the reviewer came back and said, but they didn't even look at UV. And I thought, oh my God, how could we be so dumb? We didn't look at UV, but because of the way that we collected them with our wing vouchers for every taxon that we used, we just went back and we took the wing vouchers out, with trembling hands, and it was like this independent data set that materialized up out of the phylogeny. So here you can see when we added this UV component, it was even more significant. The sister taxa that were located together were different, and when they were located apart, they were the same. Now, of course, I know you've probably immediately thought, well, there are two explanations for this. The one is just reproductive isolation and a simple differential fusion model. So new colors evolve in allopatry as a byproduct just of this divergent evolution because they've separated with each other. And only those taxa that have a high degree of a prezygotic isolation, that is, different colors to the males, will survive when they come back together in sympatry. So when they come back, that's it. But the other possibility is that this is an example of reinforcement. And the reason reinforcement is exciting is because it's one of the few areas in speciation where natural selection plays a direct role. And that is, the new colors evolve right in place of secondary contact as a result of reinforcement of reproductive isolation. So let's say the two species come together, and there were two populations have been separated. They've started to change their chromosome numbers. They come together again, and now when they mate with each other, the hybrids can no longer meiose, and so that pays a full fitness. Female pays this full fitness cost. No offspring. They either die or maybe they might be inviable. And so that's the strong driving force. That's natural selection is driving the divergence in color pattern on the wings of the males. Okay? And we had two reasons to think that this is what was going on. One is that there was actual chromosomal evidence for hybridization between sympatric pairs of these butterflies. So here you can see this hybrid between two sister taxa brought back into sympatry with each other. But the other thing that we had that was different and goes back to what Jerry Coyne and Alan Orr thought was important was we had a phylogeny, so we could put a rough date onto this divergence pattern. And the important feature that we found was that most of the pairs of species with the color differences they're not just that they were sympatric, but they also were at very small genetic distances from each other. So the differences were very recently diverged taxa, so young, these are young sympatric taxa. These are the ones that were showing the color difference. Whereas if we looked at the allopatrically distributed ones, we didn't see that early divergence. And this was our evidence in support of reinforcement. It's not completely conclusive, it's just one more step to say, you know, this data really does support this idea that it's reinforcement that's causing this pattern. And there's no theoretical problem with the idea of reinforcement. It's always been out there as a possibility. It's hard to see how it would happen. But here we had a tool, because you have an organism that has variable karyotype, and that gave us some insight into looking at this pattern. The other thing that jumped out that was really very interesting is that the sympatric sister clades tended to have a slightly higher average karyotype diversity, so they varied a little bit more in their karyotype than allopatric sister clades. Okay, so they varied a little bit more. And this suggested to us, this goes back to a regular diffusion model. This is the idea that okay, you've taken your populations apart. Now their chromosome numbers are changing away from each other. You bring them back into sympatry. If they're too similar to each other, they'll fuse, and the whole population will mix again, and you won't see them separately. But if they have to be just different enough from each other to be able to be sustained. So here their chromosome numbers are slightly changing. We test them in secondary contact with each other. The ones that chromosome numbers are too similar to each other, they meld back in, and we don't see the result of that. But the ones that are different enough from each other, so at big enough genetic distances from each other, now we see those in sympatry. And so that's our explanation for the differences in chromosome numbers. So just to kind of summarize what I've been saying, we saw two mechanisms at play here in this phylogeny of the Agrodeides. For color evolution, we think we had very good evidence for reinforcement because of the age of this group, as well as the different hues in sympatry in secondary compound. But for the chromosome numbers, we felt this was very good evidence for a diffusion model of divergent evolution. And just to pound this in once more, then there are sort of three scenarios, three evolutionary scenarios going on in this diversification of Agrodeides. There's allopatric diversification, reinforcement, and also simply allopatric speciation without reinforcement. And just in case, I know that it's hard to grasp these ideas, and I'm never sure if I explain them very well, but just to run through them again. For allopatric diversification, we separate two populations. Allopatrically, they start to diverge from each other. Chromosome number evolution, they diverge. Maybe wing pattern changes a little bit, but not very much. But the result of this is that you get these genetically very closely related, but karyotypically distinct cryptic allopatric taxa. And this is something that characterizes, I said that Agrodeides has 120 taxa, 120 species, and they've all evolved in a very rapid time period. Well, that's the result of this slow chromosome number change. Now, you can ask me, why are the chromosome numbers breaking up and changing in this way? And our answer to that is that's a question for molecular biologists. We don't know. We've looked for transposable elements because we assume that that's what's breaking them up, but we haven't, but all the taxa, both the outgroups and the in-groups, have these transposable elements. So we're still, the jury is still out on that, and we hope to do more research on that front. But for whatever reason, it does give us some insight into why you end up with separate species that look a lot alike but have different chromosome numbers. Here's our argument for reinforcement, that the chromosome numbers start to change, then they come back into sympatry with each other, and now there's very strong selection on wing color evolution driving the divergence between sister taxa that have come back in secondary sympatry. Taxa whose genetic differences are sufficiently separated. Okay, and then for allopatric speciation, this is just straight out allopatric speciation. That's also going on in this group, where you get chromosome number evolution, wing color evolution, and some other ecological traits, maybe host plants, other things going on, where here you end up, just as with all the species we're more familiar with, just with genetically distant, morphologically similar, but genetically distant species. Okay. Now, where do we go next with this? I actually, this is very, very brief, but we were originally going to work on this fantastic group that were meeting. We found a boundary where we thought hybridization was taking place. I think I showed you the slide of the hybrid karyotype that we found and so on. We even wrote a grant for NSF, which almost got funds available, not quite funded. And the next time we put it in, we made the mistake of saying where. So unfortunately, it was in Iran. And this caused terrible problems because we can't do, it's again, hard to do research there. So, and the MCZ stopped me very abruptly, and that was the end of that. After they threw general counsel at me, I kind of decided I would give up science for a little bit. But I remembered this fantastic paper by Deleuze, where he had originally done a lot of early chromosome work on Agridites, and on those three genera, the Agridites, Plubicula, and Lysandra. And the thing that fascinated me was this species Lysandra corydon. It's a very common blue you find in Europe. And every dot here is a population he sampled for karyotype. Lectureship looked at this and shakes his head and said, it's a lifetime's work. These karyotyping is hard. But the thing that was particularly fascinating is he found this very distinct cline from Eastern to Western Europe in chromosome numbers, with smaller numbers, n equals 87, being here in the West in Spain, and higher numbers here in Eastern Europe up to n equals 92. I thought, well, this has got to be a good place to start looking. And so we started collecting, and Lucas Rappel, who came to my lab, has been sequencing like mad. And I mean, without wanting to go into all of his work, he has now what I think is a good explanation for what establishes the cline, but maybe not quite as exciting. We thought maybe the karyotype itself was under selection. But actually what we think now is happening is that you get many haplotypes, much diversity here in the western part, and very few haplotypes and smaller population sizes over here in the eastern part. So basically the west is throwing off propagules that have small population size, and simply drift is taking place. And drift is causing, and since for whatever reason it's easier for the chromosome numbers to go higher than lower, what may be maintaining this cline is simply a combination of, it's simply a function of drift. The reason this is nice is because this also gives you some idea for how this phenomenon can take place of populations being separated, chromosome numbers changing, and then later, if they come back into sympatry, having reinforcement take place. So it's a nice sort of process may be revealed from this species once Lucas has finished all his work. Okay, well, to my amazement, and I guess I'm going to have to talk really fast now. We did start late, but so to my amazement, some years later, it was a centenary for Nabokov, who I think of as a great lepidopterist, but of course I know most people think of as the writer, and quite a writer, and he even published a book last year, or there was one posthumously published last year. Now I have a couple of video clips just to give you a feeling for this man. And I hope these are going to work.
I have hunted butterflies in various climes and disguises. As a pretty boy in knickerbockers and sailor cap, as a lanky cosmopolitan expatriate in flannel bags and beret, as a fat, hatless old man in shorts. I discovered in nature the non-utilitarian delights that I sought in art. Both were a form of magic. Both were a game of intricate enchantment and deception.
Okay, now what about Nabokov as a Lepidopterist? Well, you don't find a lot written about this, but Stephen Jay Gould did write a very nice and lengthy and interesting essay about the lepidoptery of Vladimir Nabokov, but it does capture the general feeling when we started this work. So no natural historian has ever viewed Nabokov as an innovator. Nabokov may have been a major general of literature, but he can only be ranked as a trustworthy, highly trained career infantryman in natural history. He divided and meticulously described. He did not unify or generalize. Now, I realize I'm already preaching to the converted here because I just put this nice, very nice story that came out of with Chris Nice and Art Shapiro's labs, and I know you all know how very much more insightful he was than this simply implies. But anyway, I'll explain why Stephen Jay Gould was not right. Okay, so Nabokov was at Harvard. He was a curator, a de facto curator at Harvard in the 40s for about, I'm forgetting if it was 6 or 8 years. He worked in the Lepidoptera room. He was a man who was well ahead of his time. He was very interested in numerical taxonomy long before this was in vogue. And here you can see he's trying to count up and think of ways to quantify the eye spots on the butterfly wings. He had what he called magic triangles, where he looked at, tried to quantify the genitalia of males to compare between species. And here, one last clip gives you a very good feeling for his feeling about the beast.
In this course, I've tried to reveal the mechanism of those wonderful toys, literary masterpieces. I have tried to make of you good readers, who read books not for the infantile purpose of identifying oneself with the characters, and not for the adolescent purpose of learning to live, and not for the academic purpose of indulging in generalizations. I have tried to teach you to read books for the sake of their form, their visions, their art. I have tried to teach you to feel a shiver of artistic satisfaction, to share not the emotions of the people in the book, but the emotions of its author, the joys and difficulties of creation. Every summer, my wife and I go butterfly hunting. The specimens are deposited at scientific institutions, such as the Museum of Comparative Zoology at Harvard or the Cornell University collection. The locality labels pinned under these butterflies will be a boon to some 21st century scholar with a taste for recondite biography. It was at such of our headquarters as Telluride, Colorado, Afton, Wyoming, Portal, Arizona, and Ashland, Oregon, that Lolita was energetically resumed in the evenings or on cloudy days. Few things indeed have I known in the way of emotion or appetite, ambition or achievement that could surpass in richness and strength the excitement of entomological exploration.
So this is a reenactment. Since Nabokov was dead when this film was, by the time this film was made. But they went out with Charles Remington, who some of you may know.
Let me look at my demon objectively. With the exception of my parents, no one really understood my obsession. And it was many years before I met a fellow sufferer.
And he would come back from a summer's collecting trip in New Hampshire, where one of the rarest of all butterflies of the East can be seen, but most of the veteran collectors won't see one in their lifetime. He saw a Strymon, Ontario, the white W hairstreak butterfly. And when he got back into town, there was no one else to talk to about it that would have been as thrilled as I was. So he immediately told me about it. And this was the kind of joy we had. The few of us who were very close to him in conversation and so on, I don't think had the slightest inkling of what he was working on in fiction. But there he was writing Lolita that captivated such a vast audience soon after. I had no idea it was coming. In fact, he seemed to me to be a rather dignified gentleman. In fact, when I read Lolita, as soon as it came out, I was astounded at the earthiness of the writer whom I knew and who was writing these things that were in Lolita.
Okay. So for his centenary, Kurt Johnson, who is himself a very interesting character, wrote this fascinating book called Nabokov's Blues, which of course I immediately picked up and started reading. And it was fantastic. It's a great read about it's the quest of science. And he pointed out in this book something I had not appreciated. I really thought that I sort of shared Gould's view that, you know, he was a very, Lycides was all there was really, and he was a very conservative taxonomist. But he pointed out that somewhere along the line, Nabokov had been presented with a box of butterflies from the Neotropics from South America. And on the basis of only about 170 specimens, he erected 5 new genera. So a new species of butterfly is unusual, but a new genus, very unusual. But 5 new genera, and said that there had been a vast radiation of these Polyommatus blues in South America that had previously been unappreciated. He gave them very nice names. This was in 1945. And so Kurt Johnson and his colleagues, Duby Benyamini and Zolt Balint, also all characters, which I could write a book about them, but they went out and they've been collecting and identifying different species, and this number has grown and grown. This was 1999, but now it's many more taxa in these different genera. And just to give you a feeling for how beautiful they are, with lovely names like Madalena and Paralycides, there's Nabokov. Somebody named this after he died for him. El Doradino, we spent a long time trying to get this one. Idalos. Okay, well, he had a very specific hypothesis that went along with this description. His idea was that these butterflies had crossed the Isthmus of Panama, come across from Asia down through North America, crossed the Isthmus, and then radiated on the Andes. But it was even more specific than that. He said that they originated here, they'd gotten all the way down and diversified here. Then all of their relatives went extinct in North America. And all these North America taxa that we see now came later in subsequent crossings across the Bering Strait. I find it easier to give a friendly little push to some of the forms and hang my distributional horseshoes on the nail of Nome, that's Nome, Alaska, rather than postulate transoceanic land bridges in other parts of the world. This was 1945, before continental drift was appreciated. So that was his comment, and there's our nail of Nome with its little horseshoe. Okay, but he did more than that. He also talked about a biologist straddling a Wellesian time machine. And so here's the actual text. Here you can imagine a point of time when the Americans had none of these Plebogani, these are the Polyommatus blues, but they were on the eve of receiving an invasion from Asia. This is 1945, so of course there's also tongue-in-cheek about the invasion from Asia. And then he said, you can imagine, our taxonomist straddling a Wellesian time machine with the purpose of exploring the Cenozoic era in a downward direction might reach a point, and then one by one, I'm not going to read the whole thing, he'll say, well, first he would see that taxa, then he would see this taxa, then he would see that one, and then that one. But of course, when I read this, I thought, oh, that's so nice. Nobody writes like this anymore. None of my papers are at all so interesting. I'll come back to this in a minute. Lovely, flowery prose. All right. Well, then enters Roger Villa. None of this work would have been possible if it hadn't been for the very lucky happenstance that Roger came from Barcelona to my lab, and he is a mountain climber. He's also a very fine lepidopterist. So here you can see, Vladimir and Roger here. Okay, and it was important because these butterflies are distributed on the tops of the Andes. This required, again, I think there were 6 expeditions in all. Some very unusual collecting techniques had to be employed at times. You would get to a moonscape on top of the Andes where there would just be little flat mats of vegetation, and lo and behold, there you would find the butterflies. So it was a lot of collecting to try to reconstruct the phylogeny. And again, I put this in here just to say we're talking about this group right here. And here's the phylogeny that came out of that. And so this is for all the Polyommatus blues. Now, why was this so exciting initially? it was exciting because agrodiitis is in the same group as these blues of the polyomantous blues that Nabokov was interested in. So we had already sampled very heavily the Old World taxa, and it was just a matter of getting these New World taxa. As you can see here, I think you can see, those of you who are used to looking at these trees, you don't need to be able to read the details or anything else. So this is the clade. This red group is the clade that he was talking about that went into South America. And right away that told us that he got it spot on, correct, that yes, indeed, these were all each other's closest relatives, and that the node that joined them was about 8 million years. That seemed about right for crossing the Isthmus of Panama and then diversifying. And there was a lot of interesting stuff going on up here with the Old World ones too. Okay, so we figured that he was correct about this. And in the Old World, too, he was correct that there seemed to be multiple invasions of North America from butterflies coming from Asia. Okay, so it's quite a few. In fact, in some cases, it seemed almost as if they were coming across, and in some cases also going back. But mostly they were coming across into North America. So just to make a long story short, we tried to date these crossings. And here you can see then that, well, now I just said about 8 million years for these ones here, but here it's estimated at 10. This allows me to say right away, we used a molecular clock, which ticked, which of course is only as accurate as your molecular clock can be, but it gives a good ballpark figure. What we knew is that this crossing didn't happen, say, 100 million years ago, which would have been an explanation if you thought it was a Gondwanan origin, nor did it happen, nor was it very close to the uplift of the Andes, which was another possibility, nor were all the taxa related, each one to an Old World taxa, which is what Kurt Johnson and Zolt Balint had previously proposed, but really did seem to follow very well with Nabokov's idea. Now, what about all the ancestors going extinct? Well, for a while I was very excited about this because, as you all know, we see this in many groups of organisms. For example, camels are always the easiest explanation. Llamas, close relatives, they're derived from camels. But of course, we don't have any camelid ancestors now in North America. Those all appear to go extinct. But we have the llamas in South America and camels, their relatives in Southeast Asia. So there are other groups that show this pattern of an extinction in North America. And I was very excited for a while by the thought that there was a bolide impact over North America that had driven the extinction of many of these North American things, because that also seemed very plausible to me as a way of driving butterflies, especially warm-adapted ones, extinct. So I was very interested by this paper, but it's become quite a controversial paper. So however that extinction happened in North America, it surely seemed to happen. And he's correct that the polyomatous blues that we now see in North America came from subsequent invasions. Okay, so here we are back at our time machine. Now, of course, we've timed our tree here using a molecular clock. Just how accurate was Nabokov? Well, he was right about the South American taxa. But then after that, remember I say he gives a list. So he then says, well, the next ones would be Ichoricia and Plebulina and the species Plebeius sepiolus. He sort of separated that one out. Well, he was correct about that timing. And then the next one, he says, the latter then would have been Lycetes agromemnon, Agriades glandon, and so on. Well, he was right also about this agromemnon for the timing of it. The next one, again, here he's got glandon. Glandon was also correct in the timing. And finally, this vaccinia, again, he was right with the timing. So 1, 2, 3, 4, 5 times in his Wellesian time machine, in the flowery prose that I thought was, oh, gee, too bad people don't write like that anymore. 5 times he was correct, or at least according to our phylogeny, he was correct in his timing. All based on morphological evidence, all based on the intuition from a deep understanding of the morphology of these butterflies. Okay, so here we have our different crossings. Now, of course, when we started to look at this, we thought, well, gee, what about, what more can we find out of this pattern? Is there anything else that we can derive from it? What about the ecology of the ancestors that crossed Bering at this time? Can we learn anything about their host plants or temperature tolerances? Well, we did some ancestral state reconstruction for that, and for the host plants, we can say that the host plants were largely, I mean, most of the ones that we know, where the life histories are known, they were also present in the New World at the time of colonization. And so we know that from Trifolium and Astragalus. Many of the host plants show patterns that are quite similar to the patterns of distribution that we see for these butterflies. And most of them are in the Fabaceae. But a more exciting approach, which is entirely Roger's here. This was his last, the last piece of putting together the colonization, because we were so interested in Bering as a corridor, was to look at what happened to the ancestors as they were crossing Bering. Because Bering, the temperature was changing at that time. As you know, during the Eocene, there was a cooling. Initially, it was almost subtropical up in Alaska. There are fossils from that time. They're almost subtropical, and then it got cooler and cooler and cooler. And that cooling is measured by the Zachos curve. I mean, so we have some very good independent estimates of the cooling. So what Roger did was he for each of these ancestors or each of these taxa that we were looking at, he got the coordinates and altitudes for the coldest and warmest locations for each taxon. This is obviously for the present day. And then he mapped their thermal range tolerances, and then he reconstructed that tolerance on the tree to estimate what the range tolerance would have been for the ancestors of those 5 crossings. So here, just very quickly, this gives you the thermal range tolerances for the different colonizers going across Bering. And when if you map them out against this Zachos curve of cooling of the water in that region, I hope you'll agree this is a remarkably good correlation. That the first crossings were very warm adapted, species that are very warm adapted, and later crossings more cold adapted. So this is. It's inferential, but it's trying to get information to really support this notion, too, of the use of Bering as a land bridge for colonizers, even for ourselves crossing, even for Clovis man crossing. It's a way to approach it. And since we had five independent crossings, we had some power to be able to look at that. Okay, well, enough on Polyommatus blues. I'll summarize quickly then, just for the Polyommatus section, the karyotype diversity. We found that it accumulates gradually in Agrodioides, that reinforcement is a likely mechanism giving rise to the speciation in the group, that in fact karyotypic diversity can be very important and directly important in promoting speciation in Agrodioides. And then finally that neotropical Polyommatus blues came from Asian ancestors that crossed Bering and dispersed in Central South America and diversified on the Andes. And one last conclusion is that I hope you will all agree with me that Vladimir Nabokov was, had he stayed in biology, who knows what he might have been able to do. But he was certainly as great a scientist as an author. I've mostly acknowledged people as I've gone along, and there are many, especially if we add in all the collectors who provided us with specimens. But here's a snapshot of the lab. It's a bit dated now, but with the main players, most of whom I've talked about already. Just to let Nabokov have the last word, I confess I do not believe in time. I like to fold my magic carpet after use in such a way as to superimpose one part of the pattern upon another. Let visitors trip. And the highest enjoyment of timelessness in a landscape selected at random is when I stand among rare butterflies and their food plants. This is ecstasy, and behind the ecstasy is something else which I cannot explain. It's like a momentary vacuum into which rushes all that I love, a sense of oneness with sun and stone, a thrill of gratitude to whom it may concern, perhaps to the contrapuntal genius of human fate or to the tender ghosts humoring a lucky mortal. So thank you very much.
Sure, we have time for questions if people can
Yeah,
leave.
people need to leave too. Thank you.
A sense with the chromosomal evolution part of this, is there evidence in insects that a mismatch of, say, 1 or 2 is a lot better than a mismatch of more, like 12 or something like that? Or is it sort of all or nothing?
Well, that can be complicated, especially, well, for these ones. For these ones, you had to have a large enough distance before it was maintained. So presumably, there's at lower rearrangement types, they fuse. So for Agrodiotes, that matters. But it's more complicated if you start to think about organisms that undergo polyploidy and so on. So I can't really say. I mean, if you have. I think it's sort of if you have a polyploid event where the whole chromosome complement is doubled or quadrupled, then you can have all kinds of interesting things going on. But those are very rare events. So, when you mean large numbers.
You know, when I hear about something that has 244 chromosomes, you know, how important can any one of those chromosomes be? And I'm wondering, I'm just wondering, is it possible that this group, that you're seeing so much chromosome karyotype evolution because for some reason, the disadvantage, because when a new karyotype arises, it's going to be, presumably, have some reproductive isolation from the population that engendered it. So you know, there's a sort of minority disadvantage problem, presumably, to overcome. Could it be, has anybody done any experimental work in this group to show that maybe there's more tolerance, less fertility cost associated with, you know, just fuse one or split one?
No, there hasn't been work, and there's nothing to tie the ecology and the karyotype yet. I mean, this is the kind of thing that I think Vladimir will continue to do. And there's a postdoc from Roger's lab is going to come to my lab who will be who's going to really look at the transposable elements, just look at the mechanism of breaking up. I mean, there are many things that you think about with this. I mean, first of all, we do find that there's more fission than there is fusion. So somehow, does that mean it's easier to form 2 telomeres than it is to fuse? You know, it raises a lot of interesting questions. Also, of course, we'd expect this to affect recombination rates. You know, there are lots of interesting evolutionary questions. As you can tell, I stumbled into this not as a population geneticist or with the genetic interest that, say, David Haig did, but, you know, having come at it more from an interest in phylogeny and these butterflies. But I suspect that there's a lot of interesting work that can be done. And we're definitely we're hoping that we can get a genome for Agrodiotes. This would be a particularly nice group to get a genome, because then we could start to look at how these chromosomes how the genome is packaged, perhaps, and which will give us some better idea about genome evolution and its consequences. I mean, correlates and consequences. Yes?
No, obviously, ecology is going to play some role in pre-zygotic isolation, but when you say there's nothing yet to link ecology and chromosome number, are you saying that you at this point don't attribute functional or phenotypic significance to chromosome number?
Sort of no, other than the correlation with that color pattern there. Just because, I mean, I guess it's my assumption that there are because in other systems it's known that transposable elements can be involved in breaking up chromosomes. So I've assumed that for some reason, I also find it very interesting that it's these three genera that show this rather strong pattern. They are independent. They're all in the same group, but they're independently evolved from each other. But it suggests that that the mechanism that's causing the breakage is not correlated with some ecological trait. So they don't have Wolbachia, or but we don't know. Yeah, it's a good question. Yes.
I'm always intrigued by possibility that sort of past kind of bottleneck events or key events can explain things that we otherwise couldn't figure out why it would be that way. So here I'm thinking of the 5 different times of coming over the Bering Straits at different times. So I wonder if you could elaborate more on whether or not, just sort of how warm it was then when they came over actually explains a lot of their ecology, their response.
Well, I think the thing that we found most surprising about this was that the thermal tolerances appeared to be conservative enough over evolutionary time that you could reconstruct them on that tree and still find what looks to be quite a good correlation between the temperatures, temperatures in the seawater there and the timing of their crossing. So that is definitely.
You can explain things like how well they're responding or how they are responding to climate change now.
Well, yeah, no, it's a good question. Actually, I think Roger is trying to sort of follow up by looking more at some of these, especially he's now back in Barcelona and looking at Palearctic species that are in really extreme environments. So it really raises a lot of interesting more questions than answers at this stage, but. Yes?
I was wondering if you could try to understand something about the history of fission and fusion of the chromosomes by looking at broad chromosomal morphology. Are these metacentric chromosomes breaking up, or are you putting extra together? Is the fundamental number constant, or does it seem like these chromosomes are getting smaller or larger?
Or are they breaking in the same places or rearranging? So that is something that Vladimir is very interested in. I don't know if you saw, but one clade in there is mostly n equals 67, and then there's another area where n is equal to 67, and he thinks that the rearrangement that gave rise to that is convergent and similar. So he's the one who's been pursuing that. He's really a geneticist, so he's been pursuing that more than I have. But yes, it's not we call it chromosome number because that's the easiest thing to count. But actually, he's interested in the karyotype, so the whole arrangement of those chromosomes relative to each other. And I do think it's going to be possible to look at that in much greater detail. But that's another reason why we're excited about trying to get a genome for the group, because we may be able to use that. We've tried fish, so I mean we've tried a number of ways to look at these, but it's so far been difficult because they're so tiny. The chromosomes are really, really tiny. It's hard to study. They're hard to study. Yeah. Yes?
I was just wondering what is happening with the host plant distributional patterns, the phytogeographical patterns in relation to these migrations.
Well, when we published, I should have put all my co-authors on that Nabokov paper, and I'm sorry, I should have mentioned that. In particular, Chuck Bell worked with us. He's interested in some of the legumes that are the host plants for these species, and they too have this rather fascinating biogeographic distribution of being in the Andes and then nothing in North America, and then in closest relatives in Southeast Asia. So there are a couple of groups in there, the astragalus and trifolium, that were actually probably here already, but then there are a few other groups that actually look like they have very similar distributions or may have had similar histories, but that's as much as we know. I mean, they had to have They had to have some suitable food when they came. Fortunately, many, and I don't know, what would be interesting would be to look at phylogenies of other butterflies that don't feed on legumes. Fortunately, the fact they do feed on legumes and probably, maybe it may have facilitated their movement. I don't know. Yeah, yeah. Well, yes?
Well, one of your early questions that you never got back to was, I believe it was Nikolai. Why did he want your students'
Oh, no. She's married to someone else, and so is he. All of them.
On that happy note.
Right. Thank you.